Take two women who are both 63 years old. One has spent her life in Japan, eating a traditional Japanese diet. The other has spent her life in Western Europe, eating a typical Western diet. Put their skin under a dermoscope. Compare the dermal collagen density, the elastic fiber organization, the fibroblast activity visible in the dermal layer. What you will see is a difference that is measurable, consistent, and has been replicated across multiple peer-reviewed studies conducted in multiple countries. The Japanese woman's skin will show collagen density and structural organization comparable to what the Western woman's skin showed at approximately 45. Not roughly comparable. Measurably comparable, quantified in published research. This is not a beauty myth. This is dermatological epidemiology. And the answer to why it exists is not what any Western skincare brand has ever had an economic incentive to tell you. Welcome to Dr. Mylin Health. Subscribe and hit the bell. Our complete anti-aging book covers every mechanism this channel investigates, the full protocol for every aspect of aging that the skincare industry profits from mice explaining. The link is in the description and in the comments. Now let me take you through exactly what the research actually shows. Before I tell you what the answer is, let me tell you what it is not, because the wrong answers are the ones the industry has been circulating for years, and they deserve to be dismissed with the data that dismisses them. It is not genetics. Studies on Japanese Americans who emigrate early and adopt a Western diet show skin aging rates that converge toward Western norms within 1 to 2 generations. If the collagen density differential between Japanese and Western women were genetic, it would survive dietary change. It does not. A landmark study published in the Journal of Epidemiology followed first and second generation Japanese Americans and documented progressive convergence of skin aging biomarkers with the Western American population in direct proportion to dietary Westernization. Genetics provides a baseline. It does not explain a differential that disappears when the diet changes. What changes with the diet is the answer. It is not the skincare products. Japan's skincare market is sophisticated and large. The country spends approximately $25 billion annually on personal care and beauty. But when researchers control for skincare product use across populations, the collagen density differential persists. Women in South Korea who use skincare products at rates comparable to Japan and at equivalent sophistication levels do not show the same differential with Western women to the same degree. The skincare products matter at the margin. They are not the explanation. It is not sun protection alone. Japanese women do have strong cultural sun protection practices, parasols, UV protective clothing, daily broad-spectrum SPF, and UV protection is one of the most well-documented preventive interventions for photoaging. But the collagen density differential observed between Japanese and Western postmenopausal women in studies controlling for UV exposure still persists after sun protection behavior is accounted for. Something else is contributing. Something that UV protection does not explain, and skincare products do not explain, and genetics does not explain. Something that disappears when Japanese women adopt a Western diet. Something dietary. Here is what the research found when it looked. The average Japanese woman consumes between 25 and 50 milligrams of isoflavones per day through her normal diet. The average Western European or North American woman consumes between 2 and 5 milligrams per day. That is a 10-fold difference in daily intake of a specific class of plant compounds that interact directly with estrogen receptors in the dermal fibroblasts, the cells responsible for producing the structural collagen that determines whether skin is firm or sagging. And that 10-fold difference is present every single day across every decade of adulthood and becomes most consequential in the decade after menopause. precisely the period when the collagen density differential between Japanese and Western women is most pronounced and most clinically significant. Let me explain the mechanism, because it is the piece of this story that the Western skincare industry has spent the most money redirecting your attention away from. Estrogen is not merely a reproductive hormone. It is a primary regulator of dermal collagen synthesis. Estrogen receptor beta, one of two estrogen receptor subtypes found in human tissue, is expressed at high density in skin fibroblasts. When estrogen binds to these receptors, it activates gene expression pathways that upregulate type 1 collagen synthesis, increase hyaluronic acid production in the dermal matrix, and suppress matrix metalloproteinase enzyme activity, the enzymatic process that degrades existing collagen. While endogenous estrogen levels are maintained, throughout the reproductive years, the dermal fibroblasts receive continuous collagen synthesis signaling through these receptors. When endogenous estrogen declines through perimenopause and menopause, which it does in every woman, regardless of ethnicity or geography, those receptors become dependent on whatever estrogenic signal they can find. And here is where the dietary difference between Japanese and Western women becomes the deciding factor. Soy isoflavones, specifically genistein and daidzein, the two primary isoflavone compounds in fermented soy foods, bind to estrogen receptor beta in dermal fibroblasts. Not identically to endogenous estrogen, not with the same binding affinity, but with sufficient receptor activation to maintain partial collagen synthesis, signaling in tissue where the endogenous estrogen signal has withdrawn. A 2007 randomized controlled trial published in the European Journal of Clinical Nutrition assigned postmenopausal women to either isoflavone supplementation or placebo for 6 months and measured dermal collagen density at baseline and study end. The isoflavone group showed a statistically significant increase in type 1 procollagen synthesis compared to placebo. Another study published in the British Journal of Dermatology confirmed measurable improvements in skin elasticity in postmenopausal women following daily isoflavone consumption over 12 weeks. The receptor mechanism works. The collagen synthesis response is real, and Japanese women have been consuming the dietary precursors to this mechanism every single day for centuries without anyone telling them it was functioning as a dermal collagen preservation strategy, because the concept of estrogen receptor beta did not exist in scientific literature until 1996. But there is a second layer to this mechanism that is more surprising and more clinically relevant than the isoflavone receptor binding itself. And it is the layer that explains why simply supplementing with isoflavone capsules, the Western pharmaceutical industry's attempt to commercialize this mechanism, does not fully replicate the outcome of the traditional Japanese dietary practice. The isoflavone daidzein can be metabolized by gut bacteria into a compound called equol. Equol has approximately 3 times the estrogenic activity of daidzein at estrogen receptor beta. It is more bioavailable, more persistent in tissue, and more effective at maintaining fibroblast collagen synthesis signaling than its dietary precursor. But equol is not produced by everyone who consumes daidzein. It is produced only by individuals whose gut microbiome contains the specific bacterial strains capable of performing the conversion. In Western populations, equol producers constitute approximately 25 to 30% of adults. In Japanese populations, equol producers constitute approximately 50 to 60% of adults. This difference was initially assumed to be genetic. It is not. It is microbial. And it is reversible. Researchers who transplanted gut bacteria from equol-producing Japanese subjects into non-producing Western subjects documented the emergence of equol production capacity within the Western subjects within 8 to 12 weeks. The bacterial capacity for the conversion can be established in a new gut environment. What maintains it? And what the traditional Japanese diet has been maintaining for 2,500 years is the continuous daily consumption of fermented foods that sustain the specific bacterial ecology required for equal production. Miso, natto, tsukemono, sake kasu. Mirin. The traditional Japanese diet is not incidentally fermented. It is systematically and continuously fermented in a way that maintains a gut bacterial environment producing equal from dietary daidzein at rates that postmenopausal Japanese women's skin fibroblasts depend on as a collagen synthesis signal. Remove the fermented foods from the diet, as emigrating Japanese Americans do when they adopt a Western dietary pattern, and equal production declines within months. The gut bacteria that perform the conversion die off without the dietary substrate that sustains them, and the dermal collagen density advantage begins to close. This is the one thing, not a product, not a topical application, not a skincare routine. A daily bowl of traditionally fermented miso soup consumed in the morning, as Japanese tradition has dictated for centuries, delivering live beneficial bacteria to the gut, dietary daidzein to the digestive system, and through the bacterial conversion that those live bacteria perform, a sustained daily delivery of equal to the circulation and ultimately to the estrogen receptor beta sites in the dermal fibroblasts where it signals collagen synthesis. 25 cents per serving. 2,500 years of continuous practice. A peer-reviewed mechanism confirmed in randomized controlled trials, and a Western skincare industry that has never once included it in a product recommendation, a dermatologist's consultation note, or a clinical skincare guide. Why not? Follow the mechanism to its commercial conclusion. An isoflavone supplement, a pharmaceutical-grade capsule containing standardized genistein and daidzein, can be patented in its formulation, manufactured in a controlled facility, sold at $40 to $60 per month, and positioned with clinical language and peer-reviewed citations as a sophisticated anti-aging intervention. Traditionally fermented miso. The dietary matrix that delivers the same compounds alongside the live bacteria needed to maximize their conversion to equal, costs 25 cents per serving, cannot be patented, has been in public domain for 2,500 years, requires no clinical endorsement to purchase, and generates no recurring pharmaceutical revenue. The pharmaceutical version of the mechanism was patented and commercialized. The dietary version of the mechanism was never promoted. That is not a coincidence. That is a commercial decision made by an industry that monetizes mechanisms it can own and ignores mechanisms it cannot. Here's what the traditional practice actually looks like, because the specificity of the dietary source matters for the equal production mechanism. Instant miso, the packets of miso paste dissolved in hot water that appear in most Western supermarkets, is produced through an accelerated fermentation process and subsequently heat treated to extend shelf life. The heat treatment kills the live bacteria that perform the daidzein to equal conversion. Instant miso delivers the isoflavone precursors, but not the bacterial cofactor required for equal synthesis. Traditionally fermented miso, aged for a minimum of 3 months, unpasteurized, and stored refrigerated, retains live bacteria at clinically relevant densities. This is the form that delivers the complete mechanism. It is available at Japanese grocery stores, Asian supermarkets, and online natural food retailers. A 500-gram container costs between $8 and $15 and produces approximately 20 servings. Dissolve 1 tablespoon in hot water, not boiling, as temperatures above 70 degrees Celsius begin to reduce bacterial viability, and consume it in the morning before or alongside breakfast. This is the traditional consumption pattern. Morning cortisol facilitates isoflavone absorption. The gut bacteria perform the equal conversion across the digestive transit period. the equol enters circulation and reaches dermal fibroblast receptor sites across the subsequent hours. Natto, fermented soybeans with a distinctive stringy texture and strong flavor, delivers the highest isoflavone concentration of any single food source. One 50-gram serving of natto contains approximately 40 to 50 milligrams of isoflavones, the full daily target documented in the clinical research, alongside a dense population of Bacillus subtilis natto bacteria that directly support equol-producing gut ecology. Japanese women who consume natto daily show the highest equol production rates and the most pronounced dermal collagen density advantages in the population studies. Natto is an acquired taste. It is also the single most efficient dietary delivery mechanism for the entire mechanism described in this video. The gut microbiome rehabilitation required for equol production takes between 8 and 12 weeks of consistent daily fermented food consumption. This is not a switch that flips immediately. It is a bacterial ecology that rebuilds gradually as the dietary substrate that sustains equol-producing bacteria is provided consistently. At 12 weeks, studies measuring equol production capacity in previously non-producing subjects who adopted traditional fermented food consumption document emergence of equol producer status in a proportion of subjects. Not all, but a measurable proportion. And in those who develop producer status, the dermal collagen synthesis signal that equol activates begins functioning at a level that postmenopausal skin has not experienced since endogenous estrogen withdrawal. The fibroblasts that were operating without an estrogenic collagen synthesis signal begin receiving one again, through a compound produced in the gut from a 25-cent bowl of miso soup consumed every morning. The Western skincare industry's response to all of this published research has been to invest in topical phytoestrogen formulations, creams and serums containing encapsulated genistein or daidzein applied to the skin surface. These products exist. They are sold in some markets as anti-aging topicals. They produce modest surface effects. They do not replicate the systemic collagen synthesis signaling produced by dietary isoflavone consumption and equol production, because they do not reach dermal fibroblast receptor sites at the concentrations required for meaningful receptor activation, and because they completely bypass the equol conversion mechanism that multiplies the estrogenic activity of the dietary isoflavone threefold before it ever reaches the receptor. The topical route was pursued because it produces a product. The dietary route was not pursued because it produces a practice. Practices cannot be bottled, patented, or sold at department store beauty counters. The 25-cent bowl of miso soup competes with $90 phytoestrogen serums at the level of the receptor and wins on mechanism, which is exactly why you have never heard a skincare brand recommend it. Japanese women are not keeping their skin firm at 60 because of their moisturizer. They are keeping it firm because their gut bacteria are converting their dietary daidzein into equol every morning while they drink a bowl of miso soup that their grandmother also drank and her grandmother before her. A practice that is 2,500 years old, a mechanism that Western science characterized in peer-reviewed literature less than 30 years ago. A dietary habit that costs 25 cents per day and that the $532 billion global skincare industry has no mechanism to monetize and therefore no incentive to recommend. Our complete anti-aging book goes deeper into every mechanism this channel investigates. The gut-skin axis, the collagen synthesis pathways, the dietary and topical protocols that address aging at the biological source rather than the cosmetic surface. Link is in the description and in the comments. If this opens something for you today, subscribe and share it with every woman you know who is spending on skincare while eating a diet that is systematically withdrawing the primary internal signal that keeps dermal fibroblasts producing collagen. The next vault opens soon. And what the skincare industry buried inside that one goes even deeper. I'm Dr. Mei Lin. Thank you for listening to Dr. Mei Lin Health. There is one more dimension of this story that belongs here, because it closes the loop on why the Western skincare industry has been so thoroughly unsuccessful at replicating the Japanese outcome, despite decades of awareness that the outcome exists and genuine scientific interest in. Understanding it. The collagen synthesis signal delivered by equol at estrogen receptor beta in dermal fibroblasts is a systemic signal. It reaches every fibroblast in every tissue via the circulation. A woman who produces equol daily and consumes adequate dietary isoflavones is providing a collagen synthesis signal not just to the fibroblasts of her face, but to the fibroblasts of her neck, her décolletage, her hands, her arms, every skin surface simultaneously. The Western skincare industry's response to collagen decline has been organized around topical products applied to specific surfaces in sequence: a face cream, a neck cream, a hand treatment, an eye serum. Each product addresses one zone. Each zone requires a separate product. Each product generates separate revenue. The Japanese dietary approach applies one mechanism systemically and simultaneously to every skin surface at once, through the circulation, every day, for the entire adult life. It does not discriminate between the face and the neck. It does not charge separately for the hands. It is not formulated for combination skin types or sensitive skin or mature skin. It is a bowl of miso soup consumed before breakfast every morning in a tradition that predates the concept of a skincare routine by approximately 2,000 years. There is also a secondary dietary mechanism that operates alongside the isoflavone pathway, and that the research has confirmed contributes independently to the collagen density advantage. The traditional Japanese diet is low in advanced glycation end products, the molecular damage that accumulates in dermal collagen when blood glucose repeatedly spikes and sugar molecules bond to protein fibers in a process called glycation. Glycated collagen is stiff, disorganized, and functionally impaired. It cannot flex, cannot transmit mechanical signals normally, and is degraded by MMP enzymes at accelerated rates compared to undamaged collagen. The traditional Japanese diet, centered on fish, vegetables, fermented foods, rice, and miso, with minimal refined sugar and ultra-processed food, produces significantly lower post-meal blood glucose spikes than the Western diet. Lower glucose spikes mean less glycation. Less glycation means less structurally damaged collagen accumulating in the dermis over decades. The collagen density advantage of the Japanese postmenopausal woman is therefore the product of two simultaneous dietary mechanisms operating in parallel across her entire adult life. One providing an estrogenic collagen synthesis signal through equol, the other protecting existing collagen from glycation-driven structural damage. Neither mechanism requires a product. Both require a dietary pattern, and both have been operating continuously in the traditional Japanese diet since long before Western science had the language to describe what they were doing or the instruments to measure the results they were producing. Start tomorrow morning. 1 tablespoon of traditionally fermented unpasteurized miso dissolved in warm water, not boiling. Drink it before breakfast. Do it every day for 12 weeks. At 12 weeks, you will have provided your gut bacteria with the sustained dietary substrate required to rebuild equol-producing capacity if that capacity was lost to dietary Westernization. You will have delivered 25 to 40 milligrams of dietary isoflavones per day to estrogen receptor beta sites in your dermal fibroblasts, and you will have begun withdrawing from the postmenopausal collagen synthesis silence that every Western woman is currently living in, not because she lacks the biology to respond, but because nobody in the $532 billion skincare industry has ever had a reason to tell her that the response requires a bowl of soup, not another jar.